I started my work more than 30 years ago when I was a young pathologist, and it was in the year of the discovery of the spirochete by Dr. Burgdorfer. And one of the key scientists who worked with Dr. Burgdorfer in that discovery, which happened to be on Shelter Island, which is about 10 miles from the hospital that I worked in, Southampton Hospital. Not to be confused with your Southampton Hospital, but it was a good Southampton Hospital. We were encouraged to gather some interest in Lyme disease, to look for it in all the areas of the hospital. All the specialists were encouraged to try to find cases. At that time, we had to make our own test reagents, so we gave a lot of time and attention to quality control so that the testing was proper and the testing was reproducible. And all of the testing that we did was certified by the New York State Department of Health under Dr. Benach's tutelage. So we got to be pretty good in diagnosing Lyme disease, both clinically and from blood testing. We made our own reagents. We took proficiency testing. We passed proficiency testing. And we were very much involved in education of all of the medical specialties, from pediatrics to geriatrics, to try to find cases of Lyme disease in their specialty. And in 1987, we had a full-day meeting, a 10-hour meeting on videotape, and each member of the medical specialties presented Lyme disease in their patient population. Pediatrics, ophthalmology, ear, nose, and throat, orthopedics, obstetrics and gynecology, psychiatry, you name it, general surgery. Every single one of those doctors on our staff had patients with Lyme disease, and they recognized it, they sent them in, the blood test confirmed, and they treated them properly. So we did a pretty good job back in 1987. And we continue to do a good job on Long Island, because Long Island is where the tick that carried the spirochete, which was discovered to be the cause of Lyme disease, that was the epicenter for Lyme in that time, in the 80s. And we also diagnosed babesiosis. I personally diagnosed 100 cases of babesiosis in all forms, crossing the placenta, causing fatalities in people who had spleens, keeping people alive who had babesiosis with no spleen, all of these other permutations and combinations. And then the other co-infections, ehrlichiosis and anaplasmosis, and all the things that came along with Lyme as a multi-infection problem. So unlike James Bond, Lyme spirochetes never travel alone. And if you have Lyme, then it's very likely that you have another infection, a co-infection, which needs to be diagnosed with appropriate testing. And so I encourage you to encourage your doctors to think not only in terms of a monad spirochete, but in terms of a microbial infectome, we call it, population of infectious organisms that travel with the Lyme spirochete. Today we're going to talk about the worst case scenario. My grandfather died of dementia. I was about 10 years old at the time, maybe 8. He was chief of ophthalmology at the Massachusetts General Hospital, Harvard Medical School. He was a brilliant man. He developed dementia. He didn't know the name of his wife. He saw hallucinations, and he was institutionalized, and he died after about 3 years. And my mother would go into the pantry, and every day she would cry for 2 hours. And I'd get home from school and she'd be in there crying. She missed him so much. We all missed him terribly. So when I decided that I was going to go to medical school, I got through and I decided I was going to be a diagnostician, not an ophthalmologist, because I can't operate on the eye. Just too much. And I decided that in my research I would do work on dementia. And I've been doing that for 35 years. And so today's lecture is the fruit of those labors. What I'm going to tell you is not generally accepted by many people in the institutional arena, and that is that there's a link between Borrelia infection and Alzheimer's disease. And I'll show you the basis of that in this slide presentation. Let's get this thing to go. So what we're really asking is, let's see here, apologize for the problem here with the, I'm going to take a picture of this and get rid of this. I'm using my left hand and I'm not left-handed. I'll have to switch over to my right, I guess. Let's see. All right. X that out. Okay. What we're really asking is dementia an infectious disease in some patients? And we can assuredly say yes, because we know that in the times past, even today, in some areas of the world, there are people who have dementia due to HIV infection, AIDS. Some people have infections related syphilis dementia, because syphilis is not eradicated from the earth. It's a public health problem in China. And late syphilis can cause dementia. It's called general paresis. Our epidemiologists, academic epidemiologists, those are doctors who study patterns of infection, are they interested in this question? Is Alzheimer's disease infectious? Well, I was delighted to read in 2013 that Dr. Mwanda and his colleagues have written an article in the Epidemiologic Reviews, which is a very prestigious Ivy League type epidemiology medical journal. Can infections cause Alzheimer's disease? So it's made the front pages of the epidemiology literature. And they're asking questions, and they're asking the right questions, because they are breaking down the infections that have some links to dementia, and they include Borrelia or Lyme, herpes simplex, chlamydia, and some other infectious agents. All of this is written in Infectious Disease Epidemiologic Reviews, and you can see here 2013. All of this will be put on the website, so you don't need to take notes. It will also be filmed, so you'll be able to see the film again. So if I go fast, don't worry about taking notes. Just listen to what I have to say, if you care to listen. If you don't want to, it's nice outside, and when the pub's not too far away. Are all dementia illnesses essentially the same? Definitely not. My mother died of dementia, but she had a vascular dementia, we think. And dementia is due to diverse causes. You can have dementia due to lack of thyroid hormone in utero, and you can wind up with dementia due to Mongolism or Down syndrome. There are lots of causes of dementia. So I'm not claiming that all dementia is infectious. I'm claiming that some of dementia is infectious, and it may be HIV, it may be syphilis, and it also may be Lyme related. Dr. Alzheimer in the early 1900s defined 2 cases of dementia, and he wrote about them. He was not well. He died fairly young. After he wrote the second case up, he passed away. Now, 2 cases don't make an epidemic, and 2 cases don't convince skeptical doctors that there is a pattern there. So from the 1900s until about 1980, nobody diagnosed Alzheimer's. Nobody. There was no Alzheimer diagnosis. It was called senile dementia, old age. And that was because when you're old, you're going to get dementia. And of course, we know that's not true today. There are many people in their 90s who have good mental function, and unfortunately, there are some people in their 90s who have Alzheimer's disease. So age doesn't mean that you're going to have dementia when you get into your 9th year or your decade or 100th year. Escalation of Alzheimer's disease in the 1980s then eliminated the category of senile dementia. Let's see what's my model. There's senile dementia. And there became a big research push to look at amyloid deposits in Alzheimer brain tissue. Also, there was a subsequent wave of interest in proteins that accumulate in the brain called phosphorylated tau proteins. These 2 camps, the amyloid camp and the tau camp, came to battle, and one assumed that that was the cause of Alzheimer's, and the other said, no, it's not yours, it's mine, and they had a tug of war. And it turns out probably neither is correct. But it is a pattern that you can see under the microscope, and it's a pattern that most pathologists use in diagnosing Alzheimer's disease. They'll do special stains for these proteins and accumulations of fibers in the brain, and they'll say, well, all right, he had mental problems, so he was demented. He has other things which I'll show you, and the stains are positive, so it's Alzheimer's disease. Now, this is from William Butler Yeats, and it was originally John F. Kennedy when he came to Dublin who quoted, and that is, some men see things as they are and ask why, I see things that never were and ask why not. And eulogized by his brother, who was also assassinated, Robert F. Kennedy, said those words, and then Edward M. Kennedy, when Robert was shot, said those words again. But they were originally written by William Butler Yeats, Nobel Prize laureate, Academy Award winner, great man of letters from Ireland. Knowledge gained and forgotten. Syphilis, 1492, Columbus. With the epidemics of syphilis in Europe, we had a major public health problem due to syphilis. And it exploded sometime after Columbus came back from the New World. So people think that he brought it with him. We don't know for sure. In medical school, we learned one night with Venus, the rest of your life with Mercury. And that was true for many hundreds of years. They switched mercury sometimes for arsenic or bismuth or other heavy metals to try to kill the bug. In the end, arsenic salvarsan came to be the most depended upon medication for treatment of syphilis before we had antibiotics. But in that time, syphilis was very virulent. It would kill people quickly and would do terrible things. It would kill babies in a great number, a huge number of stillbirths. Now, this is a kind of to paraphrase Dickens, A Tale of Two Plaques. We have the Alzheimer's disease plaques, and what they are round circles of dark staining areas. These are as if you had taken a laser and burned the brain where the black stuff is. All these things are round. That's Alzheimer's. When you have a lot of these plaques, the brain is damaged and the brain doesn't function well. So too many plaques usually goes along with dementia and Alzheimer's impairment. It's not always 100% true, because some people can have a fair number of plaques and not have dementia. So if you do the studies at the autopsy table and you find that there's a lot of staining, but you ask, was the patient demented during life, and they say no, then it is not Alzheimer's disease. It is a plaque infestation of the brain which doesn't impair cognitive function. Now, direct your attention now to C, and you can see black areas that are round in the brain tissue. And these are from general paresis, which is syphilis of the brain. And do the two look somewhat similar to you? They look somewhat similar to me. And so I'm asking for help, because pathologists never make a decision solitarily. We always ask for help. Does that look like it's could be? Oh, yes, I think it could be, yes. I mean, it definitely is. They're exactly the same. Think about it. Alzheimer's disease, plaques, dementia, tertiary syphilis, dementia, plaques. They're exactly the same. Keep it in mind. We're going to be looking at these as we go along. So William Osler, he who knows syphilis knows medicine. Now, we come into life and we experience life through a keyhole. And as we grow older, the keyhole gets bigger, and then the door opens, and then we get three-dimensional views, and we see the world, and our views change. But if you stay with peeping through the keyhole, you will learn very little about life on the planet. You'll learn very little about medicine. You will be impaired. So we commence our learning with a mere keyhole view in medicine. And we build upon that by talking to patients and by listening to patients. Listening to patients. Because everything in the medical textbook came from somebody who listened to a patient and wrote it down. And before there were patients, there were no medical textbooks. So we listened to patients. The initial keyhole view is reminiscent of Plato's allegory of the cave, and those of you who took philosophy know about Plato's allegory of the cave, where the shadows are on the wall and they can't really see what's throwing the shadows, and they have speculations about what was really going on. At inception, we had a keyhole view of Lyme disease, and that was an arthritis. And that's because it was described by a rheumatologist, Dr. Alan Steer. Cause unknown. Cause suspected viral infection. Keyhole view. Sure, swift, and wrong, right? SSW. Sure, swift, and wrong. Say that after me 3 times. And tell your GP. Sure, swift, and wrong. Lyme disease to a wider keyhole. We learned about, in addition to arthritis, we have meningitis, and the list gets better. Meningitis, arthritis, heart rhythm abnormalities. Through a wider keyhole, meningitis, arthritis, heart rhythm abnormalities, Bell's palsy, facial nerve droop, and then arthritis and erythemas of the skin. So Lyme disease now has escaped from the keyhole of the knee, and now it is involving various parts of the body, and it is almost running amok. All of these were described before we had blood tests for Lyme disease. All of these are bona fide, true manifestations of Lyme disease. All of these, when they exist in couplets, where you have arthritis and heart rhythm abnormality, Lyme disease until proven otherwise. All right? That's how simple it is. All of these were clinically diagnosed. They were diagnosed in the pre-spirochetal era. That was before we had spirochetes as the known cause of Lyme disease. All of them were diagnosed at Yale with Alan Steer's group. All of them are completely correct. What happened to clinical diagnosis? Blood tests. Blood tests. Good blood tests and not so good blood tests. And not so good blood tests have put the brakes on looking through the keyhole and seeing wider and wider manifestations of Lyme disease, right? So blood tests are not always your friend if they're not done properly. And if the materials in the blood test don't match the spirochete that's in your body and causing your disease. So we're looking through a wider keyhole now, and entered Borrelia burgdorferi, 1982. Dr. Willy Burgdorfer discovered it, and with the help of Dr. Alan Barber and Dr. Benach, they purified it, wrote it up, and it was accepted as the cause of Lyme disease, the first new spirochetal infection since syphilis. Through still a wider keyhole, we looked at blood tests. And let's see if I can get this thing back here. Blood testing for antibodies. Now, blood testing for antibodies can be helpful if it's done properly. If the blood test is negative, the blood test does not say that you do not have the disease. In immunopathology, only positive results count in immunopathology. Repeat after me. Only positive results count in immunopathology. Negative results could mean you did the wrong test, you had a bad batch, you didn't know what you were doing, you got a negative. It's always good to get a negative. I could reach out the window and grab some air and pull it in, and that's a negative. It doesn't help you with your diagnosis, does it? Through still a wider keyhole, we had some validation by blood testing. And then we have the 4 signs of Steer. Mankind, now is mankind prepared to live with a mere 4 categories of Lyme disease, like it's in the textbook? Are we content with the small keyhole view of Lyme disease, or are we going to allow it to expand into other areas of the body, like the eye, uterus, the ear, the deep brain? Vienna, Austria, 1985. I presented a series of things that should not have existed under Dr. Steer's 4-point Lyme disease paradigm. I presented dead babies, babies that had been infected in utero. I did an autopsy on the babies. I found spirochetes in the baby's autopsy tissue. I sat and I showed in 10 minutes 100 slides of spirochetes in dead baby tissues. And at the end of my talk, the jaws had dropped, and nobody asked any questions, and they quietly walked out of the room, and they ignored it. 1985, Vienna. Codification. We learned that We have primary Lyme, just like primary syphilis. We have secondary Lyme, just like secondary syphilis. We have tertiary Lyme, which is late after the initial infection. And those are how we classify, in one sense, the stages of Lyme disease: primary, secondary, tertiary. In Vienna, they said at the time, 1985, tertiary and Lyme in the brain. And I heard tertiary and Lyme in the brain, and I had a lightning bolt arc in my head: tertiary syphilis, dementia; tertiary Lyme in the disease in the brain. I wanted to get on a plane and go home and get my hands on some Alzheimer tissue. So we had some reconciliation with the European observers, but they were still not on board with the tertiary manifestation of dementia. They were on board with acrodermatitis chronic atrophicaans, which is a skin infection. That's tertiary. Banwarts, which is painful radiculoneuritis, a pain in the nerves, secondary. And Borrelia lymphocytoma is secondary. Fetal autopsy data showed that the babies had indeed died of Lyme disease, and their tissues were riddled with spirochetes. So human fatalities: fetus, fatal, shocking. Lyme disease is not supposed to kill. Lyme disease is not supposed to kill dead babies, kill babies. Truly shocking. And because it was shocking, it was ignored. This is one of the babies. This is a heart. This is a small area of the heart, and all these things here are spirochetes. I've split them out so you can see how the spirochetes stand out. And those are all spirochetal infection in the baby's heart. We introduced the lab culture, and from those hearts we grew pure cultures of Lyme spirochetes. So not only did we have the spirochetes under the microscope, we had the spirochetes growing from the diseased but dead heart in the test tube. So we had two ways of proving that the baby had Lyme disease. Tertiary is a motif for all spirochetal infections. Well, in tertiary syphilis, it's okay. That's in the textbook. Tertiary Lyme disease, it's okay if it's in the skin, not okay if it's anywhere else. Now, I dispute that. I say tertiary can wind up in the brain and do to the brain what syphilis could do with the brain when tertiary syphilis got to the brain, that is to cause dementia. That does not require an IQ of over 80, right? My IQ used to be higher, but I think it's somewhere around 80. Even I can get that. So, tertiary borreliosis in the skin, bulletproof evidence. There's no contention about it. European scholars, acrodermatitis, implications of tertiary, spirochete, no problem. Syphilis, dementia. Syphilis, bulletproof evidence. Brain, spinal cord, heart, fetal, infant deaths, all terrible, terrible consequences of syphilis. Lyme can do every one of those things. Everything that syphilis can do, Lyme can do. So here is a confession from me. Everything in my scientific career I stole from syphilis. I stole it. Stole it outright. Problem was there were no owners around because all the people who owned those concepts had died 500 years previous. And nobody today cares in the way that I care about tertiary spirochetal infections, especially in Lyme, and especially in syphilis. I've read every textbook on syphilis in the English language. I don't read German, although otherwise I would have read Dr. MacDonald's textbook, which is that thick. So my hypothesis was that tertiary infections might produce dementia. A bold move. And I got brains frozen from an Alzheimer brain bank, and I confirmed that spirochetes, Borrelia spirochetes, not syphilis, were in every single one of the brains under the microscope. And just like with the babies, I grew the spirochetes from every single one of the brains. You can't grow syphilis spirochetes in the test tube. So in Lyme disease, you got more proof than syphilis could ever hope to show that the spirochetes are the bad boys. Evidence-based medicine for Borrelia-induced Alzheimer's, 1985. Journal of the American Medical Association. I have published in journals where there is no fee assigned to me to publish. This is like the New England Journal of Medicine or the Lancet. This is the same quality. Journal of the American Medical Association, 1985. I put it on the map. These are the spirochetes. I grew them out of Alzheimer brain. These are the spirochetes in the brain. They look kind of wiggly like worms. This is a cystic form. I put the cystic form on the map. Nobody prior to that had diagnosed a cystic form of Lyme disease. This is like a bag of water. And because it's lost some of the water, it's wrinkled. These are the wrinkles. But when it was full, it didn't have wrinkles. So this can become this, and this can become this, and this can become this, and they are all interconverted. Alzheimer brain tissue. So we have a tabulation of medical publications now just completed, and I call your attention to the free website vimeo.com. And I have tabulated 126 autopsies of Lyme worldwide from many different languages after reviewing 6,000 published articles. We come up with 126 bona fide documented Lyme autopsies. Evidence point number one, pure culture. No contamination. That takes a lot of work. Your NHS guy is not going to be able to do that for you. Your microbiologist is not going to be able to do it for you. Dr. O'Connell never did it. Never. Right? And she was the Lyme czar here in the UK for many, many years before she retired. Pure cultures of Borrelia are hard to do. Culture from autopsy tissue is even harder. I have a stable full of these. Evidence point number two, monoclonal Borrelia-specific antibodies to prove the spirochetes are Borrelia. There are specific antibodies which will react with Borrelia and nothing else. That's what I used in my repertoire from Dr. Alan Barber. Validation studies, Alzheimer Borreliosis. Now I'm using the two words together. I'm boldly stepped forward and combined Alzheimer with Borrelia infections when I grow the bug from the dead brain and when I can see the bug under the microscope in sections from the dead brain. Dr. Miranda and I in 1987 published another case, and this is in Human Pathology. This is an academic journal in the US. Professors of pathology publish in this journal in the US, and it's recognized worldwide, 1987. Again, no fee to publish. That was published on its own merit. I had to beat out several hundred other articles to get that thing published. And it was judged to be qualified for publication in that very exclusive academic journal. And then the New York State Annals of the New York Academy of Sciences, that speaks for itself. Another report, this is where I described for the first time the spirochetal cyst form, which I showed you earlier, also in the case of Alzheimer's disease. Neocortical borreliosis means the infection is up here, the front of the brain. Alzheimer's disease speaks for itself. Spirochete, that's Lyme disease. This is the cystic form that I found. Notice it's wrinkled again, remember? It used to have more fluid like a balloon, lost some fluid in fixation, became more wrinkled, all right? And this is what the cyst form looks like when you roll it out like a fingerprint preparation. This is the cystic form using the monoclonal antibodies. Alzheimer's disease. Now, from the period of 1985 to 1993, nobody in the world was doing anything to try to duplicate the studies that I had done earlier. And some people said that I had done work that was not to be trusted, that I was fraudulent, that I had created data out of some fraudulent source, until Dr. Judith McCloskey, there she is. Hello, Judith. Thank you, Judith. Judith came to my rescue. And she duplicated each and every one of my observations. And we didn't talk to each other before she published her paper. She had 14 cases in her initial paper. And when Dr. McCloskey published, they stopped calling me fraudulent. They might have called me other names, but It was sure now that a second qualified academic professor of neuropathology in Switzerland could find the same things that MacDonald was finding in Southampton Hospital in Long Island, New York. Positive cultures, positive spirochetes in brain, Alzheimer's disease. Same thing. Those two steps are called validation. So you may say, all right, I have an idea, I've worked through it, I've come up with a better recipe for kidney pie, and nobody tastes it. And then you get validated when somebody from across the continent, maybe in Edinburgh, comes up with the same formulation and says, this is the best recipe for kidney pie I've ever had. And they are validated. One validates the other, working independently. They come up with the same conclusion. That's what validation is in science. And ideally, validators should not know each other, should not work together in the same lab, should not be good friends, should just be academically focused on doing the science and repeating the experiment. So Dr. McCloskey found that the plaques of Alzheimer's were positive for Borrelia proteins. I had found that too. Dr. McCloskey did DNA studies. I had done that too. And Dr. McCloskey found that the cultures were positive for spirochetes. I had done that too. That's validation. 3 points of validation. 2 points determine a line, 3 points determine a plane. 4 points is Carl Sagan stuff. All right, validation of Borrelia-induced Alzheimer's disease using molecular beacons. Now, beyond doing stains, which is protein-based research, you can do DNA-based research. So I've moved over now to molecular beacons, which are DNA probes, and they seek out only the Borrelia DNA, and when they find it, they latch onto it, and they interrogate the tissue, all right? And if the probe sticks to the tissue, then you have a positive interrogation. The beacon DNA probe finds Borrelia DNA in the tissue, and that tells you that no matter what form the Borrelia has, is it cystic, is it spiral, or is it granular, as about to see, it's still Borrelia DNA. And it still does not belong in human tissue, period. So this is where the action is now, 21st century molecular beacon stuff. I've also used PCR technique to amplify Borrelia from Alzheimer brain tissue. I've digested it, then done this amplification with the polymerase chain reaction method, and I'll show you some results here. We have sharp bands, 1, 2, 3, 4, 5, 6, and there's a very faint one up here, 7. Each one of these is specific target that I designed to be amplified out of millions and millions of possible DNA targets. I wanted a Borrelia target, so I designed primers to amplify just these bands. See how sharp they are? I cut these out of the gel. I sent them to a world-class DNA sequencing lab. They extracted the DNA out of the gel, and then they sequenced the DNA. So DNA is a series of letters, A, T, G, C, et cetera, et cetera. And I got the results back. And each and every one of these on PCR amplification had Borrelia DNA in these bands, exactly as I had designed it. There's also another piece of the story, but I won't get into that now. So polymerase chain reaction, another way of diagnosing Lyme disease. This is a bad PCR. You know, you've heard of sloppy work. Well, when I first started, I was trying to get something nice and sharp. And I wound up with stuff like this. These are smears, right? Skid marks. Worthless. Can't diagnose anything on that. So it took me a matter of probably 7 months to go from this to the sharp bands. And I just deposited all these into the Harvard Brain Bank and the National Gene Bank and positive for Borrelia by DNA sequence analysis. That is the most precise identification technology available today. You can go to the electric chair based on DNA evidence. If they find your DNA at the crime scene, you're toast. All right? That's how good DNA is. When the plane crashed into the World Trade Center and they had to identify people by little itty-bitty pieces, they used DNA technology to identify people. So your DNA is your fingerprint, and we all have our own DNA fingerprint. Borrelia is the same way. There's the sharp bands again. All of them sharp, all of them had the intended target, all of them pure Borrelia. Nothing in the human genome has the DNA sequences of these bands extracted from Alzheimer brain tissue. So I'm now advocating DNA probes to localize spirochetes. Alzheimer's under the microscope, we have plaques. In addition to plaques, we have tangles, and we have these granular things called granular vacuolar degeneration. So here are the plaques, the burned round spots. Now think about it. These are round. What did the syphilis brain look like? Round. What did the Alzheimer brain look like? Round, right? Same deal. These are tangles. They're like threads, extruded fibers. Not normal in brain tissue, but present in Alzheimer's disease. And these are granules. Now I want to pay attention to these granules because I love these granules. I'll show you where they come from. This is something that you see in Alzheimer's disease. 1, 2, 3. Plaques, tangles, granules. Granules are inside the cell. This is the nucleus. They kill the cell from the inside out. Once you've got granules in your neuron, kiss it goodbye. Neuron is dead. All right, plaques. Plaques in Alzheimer's disease. I use my DNA probes to look at the plaques of Alzheimer's. That's what an Alzheimer plaque looks like with my DNA probe. All the green is the DNA probe sticking to the Alzheimer plaque. There are holes. We'll talk about them. This is jet black. This is the adjacent brain tissue. This is Borrelia DNA. No Borrelia DNA here, 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 or here. All here. Tangles. Disease microtubules? No, I don't think so. We'll get into that in a minute. Granular vacuolar degeneration. These are granules, and they're inside a bubble. You can see the granule and then the bubble. Once they get inside the cell and infiltrate the cell, they kill it from the inside out. Spirochete can break up into little pieces, like if you have a pearl necklace and you unstring the pearls, you get things like this. So this is a pearl necklace, and these granules are the pearls unstrung. Each one of those granules has all of the DNA necessary to remanufacture this or to make a new cyst. So it's a multiplication effect. One spirochete can produce 30 granules if it's short, stops here, or 60 granules if it's long. So you multiply the infecting ability of one of these 30 to 60 times, and you get those granules, which are all infectious. And you know, here they are beginning to form in an old spirochete. It's breaking up into little sausage-like and pearl-like things. Those will be released. Each one of those has all the DNA to remake the spirochete or the cyst. DNA probes. The DNA probe here is red. What do you see? Red round things inside the cell. Not the nucleus, inside the cytoplasm. The nucleus is here. These are the granules. This is the Borrelia DNA probe. This is human. This is Borrelia. Borrelia, human, Borrelia, human, Borrelia, human. That cell is toast. This is with the green stain. Nucleus is over here. These are so many granules that are overlapping. Cell is toast. This is a cell with just a few granules. 1, 2, 3, 4. Maybe 5 out of focus. All of those are DNA probes, Borrelia only. The rest of the cell is dark. Now the tangles I used to think were carcasses of neurotubules. But Dr. Kidd from the UK proved me wrong, others wrong, and he has defined an architecture to the tangles, and they're more filamentous. So it's a fiberopathy, an abnormal fiber. And to this day, I don't know what causes tangles. I can't connect it back to the Borrelia yet. Dr. Judith McCloskey, here she is again. Judith, thank you so much. Using DNA probes, DNA analysis, to localize Borrelia to the sites of Alzheimer's disease tissue injury. So she's using the same DNA tools in a different venue in Switzerland to prove with DNA tube technology that the Borrelia signature, DNA signature, is in the Alzheimer tissue. She uses immunoblots. She finds bacterial cell wall in Alzheimer's plaques. She uses culture. She gets pure cultures. I got pure cultures. And she does interesting experiments in the test tube. This is a test tube which was combined healthy cells, nerve cells, single cells in tissue culture with a whole bunch of Borrelia growing madly. And what happened was the Borrelia, let's see where it is, here, is inside the cell. So it penetrated the tissue culture cells from the outside in, and there it is inside the cell using a specific DNA marker. This is one of my works. This is a nucleus. This is a Borrelia inside a nerve cell from an Alzheimer case. Dr. McCloskey, she's also used atomic force microscopy to show the cystic forms that we, round forms we showed you before, all in Alzheimer's disease tissue. Now, we're going to get into a little bit about the holes. You can see that the amount of glow is a little bit uneven, and the cyst has DNA-rich areas, and then it has membrane material around it, which is DNA poor, but the DNA nonetheless. Dr. Shoppy and I have investigated something called biofilms. Biofilms are communities of Borrelia that are like a city, and they have specialization of function and specialization of shape, and they work together for the greater benefit of the community and keep the community from being wiped out by harm. This is a Borrelia biofilm. So you'll recognize here as a spiral form. Every one of these other dots is also a Borrelia, but a granular form. Those little dot-like things, right? And this material in between the spirals and the dots is like a glue that holds it together. It's called a matrix. And it's formed from the bodies of once living, now dead Borrelia. This is a biofilm in acrodermatitis chronica atrophicans, a chronic skin disease in Europe. We don't have it in the US. Notice that these are all dot-like things. These are the granular forms. This has probably been sitting in the patient's skin for 30 to 40 years. They don't stay spiral. Over long periods of time, they evolve and metamorphose into other shapes and sizes. This is a biofilm, and you can see granules, you can see cysts. My phase contrast study then shows cysts and granules. I don't see a single spiral in there. That's a perfect Borrelia biofilm raised in a test tube. These blue blobs are biofilm in the tick gut. So when the tick feeds on you, you may get, instead of one spirochete into your blood, you may get a whole hunk of biofilm into your blood. A larger infection unit. This is an Alzheimer plaque. This Alzheimer plaque from La Jolla, California, Scripps Institute, shows a round thingy, and then it shows these thingies. And this whole thing is round, and this is the healthy brain tissue around it. And then there are these empty spaces I was going to tell you about, and I'm telling you about them right now. See, they're empty, they're black. Those are water channels. That's the septic system and the nutrient system to feed the biofilm community. These are channels like streets in London, curved, allowing water to get from the outside in, and nutrients to get in, and waste material to get out. Water flows through these spaces and allows nutrients to flow in and waste to flow out. So these holes in this Alzheimer plaque are water channels. And water channels are the sine qua non of a community of microbes, all sharing work, having a sanitary engineer designing these things. You have sewers in London, right? Biofilms have sewers. And that's what it is. So, Alzheimer plaques and Borrelia biofilms. My hypothesis is, based on DNA probes, that these brown things are actually these biofilm communities. Biofilms are always round. They come in small, medium, and large. Alzheimer plaques are always round. They come in small, medium, and large. They have water spaces. They bind Borrelia DNA. These are communities of Borrelia forming round biofilm units, destroying the human brain in Alzheimer's disease. Biofilms are, to my way of thinking, the unifying concept to explain why you have small plaques, medium plaques, large plaques, and they're always round. And they always have these empty spaces. And they look just like biofilms reared in the test tube that Borrelia does. This makes Alzheimer's with Borrelia an infectious disease, but worse than an infectious disease of the ordinary type. It's a biofilm infection. And biofilm infections are notoriously hard to eradicate. My wife just had her hip replaced. She doesn't have an infection in the hip. God forbid that she should. If her hip ever becomes infected, all the antibiotics in the world will never get that infection out. They'll have to take her to the surgery, remove the infected hip, rip it out, give her time to recover, and put in a clean one. Because once biofilms grab you by the throat, they never let go. Unless you have a biofilm treatment protocol, biofilm busting protocol. And we can get into that a little bit in the question. So here are the final parting shots. These are spirochetes. If these are the only spirochetes, you get marked wrong, because they also look like this. Those are spirochetes. And they look like this. Those are spirochetes. Can you connect the dots? Sure, you can connect the dots. These are DNA, RNA strands, and where the break point is where these things break apart. And then a cylinder, which is like a sausage, in free liquid medium assumes, according to which law, that sphere is the most efficient mathematical entity to have a relationship between surface area and volume. Sausages become round. So these sausages will become the big round plaques, and these little guys will stay round. That's why the plaques are a different size. Now, what I presented to you is the DNA probes are the best stains on the planet Earth for Borrelia detection. And in a parting shot, taking a fresh Alzheimer brain and doing a fingerprint preparation and then hitting it with a DNA probe, boom, what's that? With a DNA probe, specific for Borrelia. That's a spirochete that's just rolled out of a fresh fingerprint preparation of autopsy human brain. So with this, we shall conclude. This is the young, ferocious MacDonald, 1987. This is the senescent, retiring, just so pathetic. And 2013. Thank you very much for your kind attention. you.